All right, so I'm going to pick up where I left off in the last lecture on the cellular biomarkers or cellular hallmarks, if you will, of brain aging. And I pointed out this reciprocal relationship between the mTOR pathway and autophagy. So the point here was that factors that activate mTOR tend to inhibit autophagy. So some of those factors are growth factors, such as insulin-like growth factor 1 and others. And then amino acids in particular were called branched chain amino acids. These are leucine, isoleucine, and valine. And so these will activate mTOR, and that promotes, kind of beefs up overall protein synthesis and puts cells in a growth mode. Um, And so the way that the autophagy pathway is inhibited is that mTOR inhibits this ubiquitin-like kinase 1, ULK1, which is an important upstream stimulator of autophagy. It recruits these ATG autophagy related proteins, and you get these protein interactions and recognition of cargoes, whether it's abnormally folded proteins or uh, damaged membranes or even organelles. And this LC3 protein is very important in recruitment to autophagic membranes and formation of a autophagisome, which then fuses with the lysosomes. Okay, so anyway, the, the next few slides, I'm going to talk about some experiments done by a group at University of Wisconsin, Dudley Lamming, who's manipulated these branched chain amino acids in the diet of mice, both wild type mice and a mouse model of Alzheimer's disease. And they've looked at aging and a bunch of different indicators of general health, metabolic health, and so on. Right, and this is kind of a summary of this actually. The idea is that these branched chain amino acids activate mTOR. So if you uh, reduce dietary availability of this will, from the standpoint of the mTOR pathway, be kind of like uh, uh, fasting in a sense that you get reduced mTOR activity and cells kind of go into a stress resistance, conserve resources mode. Um, okay, and I also mentioned, I think it was the last slide of the last lecture, that there are lifespan studies in mice, three center studies, who collectively found that rapamycin, which inhibits mTOR pathway, can extend lifespan. And also in those studies, they've looked some at brain health with the rapamycin study and seen improvement. Okay, so in the next few slides is a study that, oh my gosh, didn't I cite? I cited here, it was in Nature Aging last year, one of the recent additions to the Nature collection. All right, so what they did is they started with kind of middle-aged or even older mice, actually 20 months, which is getting old in mice. And then they did 4 months. They switched them. They divide them into four groups: control diet, uh, low isoleucine, low amino acids, all amino acids, and then they had young animals on the normal diet as a comparison. And So what they found was that the animals with low total amino acids or isoleucine, they had lower body weights and they had improvements in glucose regulation. This is a glucose tolerance test where you give the animals a bolus of glucose and blood, and then they're measuring blood glucose, which goes up and then um, comes down over a period of a few hours. And what they found is that there's a less of an increase in blood glucose and a faster recovery towards baseline levels. And here's kind of the, I guess, the 120-minute data. And they also looked at other things like energy expenditure. Um, uh, so essentially, this dark is the night, say 12 hours of darkness where the animals are active. That's their nocturnal, that's their active period. So their energy expenditure goes up because they're moving around a lot around in the cage. And then they looked at this fibroblast growth factor 21, FGF 21. There's a lot of interesting work on showing correlations between circulating levels of FGF 21 and kind of youthful states in general, good health. And they found that the low isoleucine and low total amino acid diets elevated FGF 21. Um, okay, so, and then they looked at some other measures of function of the mice, including brain function. One was like a frailty score, which is it's a collection of uh, phenotypes that this one group put together and called it a frailty score. And so on the low isoleucine and low total amino acids, there's a reduction in the frailty score. And this is the young animals here. And then they did rotorod tests. So you put the mice on a rotating rod, and usually you, over time, increase the velocity of the rotation. So it's like standing on a log in the water trying to roll it. And at some point, it gets going fast enough, they fall off. So that's rotorod time. And so the aged animals have, oh, sorry, got to look back at this. So this is aged. This is aged low isoleucine, low total amino acids, and this is young. So anyway, these three conditions, the animals stay on the rotor rod longer. And then they also looked at their ability to cling to essentially a rope. And so the young animals were able to hold on longer than all the other ones, but the ones on low isoleucine um, actually were worse. So the strength here seemed to be not as good with the low isoleucine. Then they did Barnes maze, which is this board with holes in it and essentially food rewards in the holes. And the animal, it's set up so the animal has to uh, remember where the food reward was. And this measures the time to get to the right hole. And the animals on the low isoleucine, sorry, low total amino acid did better than the aged on the normal diet. And the low isoleucine did not have a beneficial effect on the memory in that test. Oh, sorry, this was males, and they did both males and females. Overall, pretty similar results with some, I guess, quantitative differences between males and females. Okay, then the second study was using a mouse model of Alzheimer's disease that was developed by Frank LaFurla with my lab's help. There's a certain inherited forms of Alzheimer's disease. I'll talk about this the next lecture, that they're caused by mutations in either the amyloid precursor protein, which is the protein from which the amyloid beta peptide that accumulates in plaques in Alzheimer's disease comes from, APP. And then, but there's even more families that have been identified with mutations in the gene presenilin-1. And the presenilin-1 gene encodes a protein that cleaves the amyloid precursor protein to liberate the 40 to 42 amino acid amyloid beta peptide that self-aggregates in the brain in Alzheimer's disease. And we, with the help of George Martin at University of Washington, had made and characterized presenilin-1 mutant knock-in mice. So they have this missense mutation that causes early onset Alzheimer's disease. And we knock that exon with that mutation into the endogenous mouse gene, presenilin gene. So the point here is then the expression of the mutant presenilin-1 is under the control of all the normal transcriptional regulators. And that contrasts with kind of the old school transgenic mice where the transgene is kind of incorporated randomly into the genome and is constitutively expressed. But anyway, we sent him these mice, and they bred them and got female pregnant and took out the like 1 or 2 stage embryo. And they injected with 2 different expression plasmids, one to express a mutant form of tau that causes frontotemporal dementia, which is a disorder where there's a lot of neurofibrillary tangles, particularly in the frontal cortex and temporal lobe, but little or no amyloid accumulation. And then also a construct with APP mutation. So then when this mouse grew up, it had all 3 mutations. So that's why it's called triple transgenic model. And as they get old, they get a lot, and this is from my lab, a lot of A beta plaque-like accumulation in the cortex and hippocampus. And then this shows hippocampal CA1 neurons immunostaining with human tau antibodies. So these are the tau and this APP are human. One thing I should point out, so these presenilin-1 mutant knock-in mice, we didn't find that, first of all, they have no amyloid pathology, no tau pathology as they get old, and they have negligible cognitive deficits, although interestingly, we did find neurons in these mice, for example, in the hippocampus, show increased vulnerability to excitotoxicity, and we did a bunch of studies suggesting that has something to do with the role for this presenilin-1 protein in regulating calcium levels in cells. Okay, so anyway, and we'd also shown actually doing functional MRI in collaboration with a group at NIDA, National Institute on Drug Abuse, that in multiple brain regions, these 3xTGAD mice have hyperexcitability, essentially, something that's called, we call, not we, others had called regional synchronicity within a brain region. And there's reduced connectivity between brain regions. Okay, so the group of Wisconsin took these mice uh, and they put them on either control, isoleucine restricted, leucine restricted, or valine restricted. So each of the four branch chain amino acids that are known to activate the mTOR pathway, and they looked at the um, Barnes maze and novel object recognition, and they found that the what did they find? The valine and isoleucine restriction in particular, there was some improvement and somewhat too in the valine restricted animals compared to the control diet. And this is quantified here. And then in the novel object recognitions, where the animal mouse has to remember what objects were in the cage previously, and then you take them out of the cage, put them back in later, but you replace one of the objects with a different object and see how long they will explore the new object, the novel object, longer time than the old one. And anyway, there was the results here for short-term memory. There was a clear effect of all three amino acids restriction, and with the long-term memory, only with the isoleucine. Right. And this is just more data. This is in the females, the previous was the male. Okay. Yeah, I'm going to skip that. And they also looked at the brains and looked at the amyloid and tau pathology and found that, and this was interesting, the isoleucine and leucine restriction reduced A beta plaques. The valine restriction didn't reduce, it even increased A beta plaques, but on the other hand, all three amino acid restrictions reduced the tau pathology. So one interpretation of these findings is that um, that the amino acid restrictions and maybe through inhibition of mTOR is protecting neurons against neurotoxic effects of amyloid beta peptide. And actually, there's a lot of evidence from this from humans. Some individuals who are in their 90s are still cognitively sharp, and their learning and memory is fine, but at autopsy, they have a lot of amyloid plaques in their brains. Okay, so now the rest of the lecture is going to be on just some data in humans, mainly on functional changes in brain during aging, structural changes. So the functional will be mainly psychological tests and functional MRI. And then the structural is either MRI or postmortem examination in some cases. And then I'll also talk about at the end about biomarkers of aging and brain aging, which is a really hot area now, doing a lot of analyses of brain tissue, cerebrospinal fluid, blood, um, transcriptomics, uh, proteomics, metabolomics, And I'll briefly mention new methods for isolating small vesicles that are probably, well, we know they are released from neurons and other cells, microvesicles or uh, exosomes. And these can find their way from the brain to the blood. And actually, it's possible now to isolate all of the vesicles in the blood, which are coming from all sorts of different tissues, heart, liver, whatever. But using antibodies against neuron-specific proteins, it's possible to pull out from that total pool of vesicles those that are likely coming from neurons, although I'm not sure it's the brain. It could be peripheral neurons too. But anyway, I'll talk about those at the end. Now, there's a lot of cross-sectional studies. So just comparing individuals at different ages and measuring something or testing some functional aspect of the brain. Then there's longitudinal studies in which you follow the same individuals over periods of decades. Usually they start with enrolling the subjects in the studies when they're middle-aged, some even younger. One of the most famous one was a Framingham Heart study, which was a study that first showed a correlation, an association between elevated cholesterol and cardiovascular disease, atherosclerotic cardiovascular disease. Then one here in Baltimore longitudinal study of aging, National Institute on Aging. Actually, a lot of Hopkins investigators take advantage of data from this study too and collaborate. And then there's studies that just uh, they're called centenarian studies. So they're studying people who live to be 100 years or more and comparing various aspects of their lifestyle, diet, and genetics to individuals that don't live so long. And there's also some studies of particular populations throughout the world, one in Okinawa, one in Sardinia in Italy, where there seems to be exceptional longevity. Um, the lay public people may have heard of the blue zones. So these are these areas in different regions of the world where people living there live a long time, and it seems the data suggests it's not, most of their increase in longevity is not genetic. Okay, and then twin studies, kind of the classic things, comparing identical twins and who are living under similar or different conditions. Okay, so just some kind of simple things on decline in function in general with age. So these are the half marathon world record times. Well, I guess some are world records and some are whatever USA records, it doesn't matter. The point here is that for endurance running anyway, you peak when you're in your 20s and 30s, and then by the time of 40, the times go up and up and up with kind of a exponential increase in times, indicating an exponential decline in performance. VO2 max goes down with aging in both men and women. With that trajectory, it kind of follows closely the decline in endurance performance, which makes sense. If we look at sensory systems, vision declines with aging, hearing declines with aging. It's actually pretty dramatic. You can go on the internet and get these, test your own hearing, particularly ability to detect high frequencies. And I did this with my kids. Well, they aren't kids anymore. Like they're in their 20s. They were in their 20s and maybe 10 years ago. And yeah, they could hear things at high frequencies that I couldn't hear. Okay, so walking speed declines with aging. And all these things, there's a scatter, right? So a lot of inter-individual variability. Uh, and it's important to try to understand that variability. And there is work going on that. Um, Essential tremor, which is it's kind of a benign tremor that people can get as they age, but the incidence goes up with aging. Um, Ability to regulate blood glucose levels declines with aging. So increased insulin resistance and diabetes with advancing age. And the immune system declines with aging. This shows changes in the thymus gland, which actually shrinks a lot uh, beginning um, like at 1 year of age, actually. Uh, so, kids, when they're young and are exposed to infectious agents, um, their immune system is really good at being able to detect those pathogens later in life, but there is some waning of the immunity. Right. So we all know this. Older people are more susceptible to bad outcomes with various infections. And this just shows looking at some immune responses to different specific antigens, phytohemagglutinin, concanavalin A, pokeweed mitogen. Um, all right. Okay, so the brain in humans, uh, most cognitive domains decline with aging. So this just shows reasoning, uh, processing speed, and memory. But again, there's a lot of variability, and it's important to understand that variability. Some of it definitely has to do with the environment, diet, lifestyle, keeping the mind intellectually and socially engaged as you age, and keeping a healthy body weight, getting physical exercise. I'll talk about all that stuff in my last lecture. This is from a review article looking at this again now. Um, what doesn't decline with aging is verbal ability. So your vocabulary doesn't really decline. It's mainly being able to process information quickly and to remember new things that you learn. Okay, and general, at least basic math skills, being able to add, multiply, divide, and during normal aging, not much decline, but these other domains. One thing that declines a lot is being able to switch your attention between different tasks and, be focusing on one task and then quickly get your thoughts off that task and engaged with another task. Um, yeah, so this kind of idea of fluid intelligence declines and these domains decline. If you're interested in details on this, you can read some of these. There's a big literature on it. Okay, so in animals, I'm switching back to animals a little bit, where you have genetically, or not always, but often genetically homogeneous animals that are housed in the same environment, and you compare young and old animals, or you look at them longitudinally, look at changes in individual animals over their life course, and then you can manipulate precisely different aspects of their environment. You can obviously do learning and memory tests. You can interrogate the brain at the histological, cellular, molecular levels, and you can do genetic manipulations and so on. Um, this is from, yeah, this is from Peter Rapp's group. What year was it? 2016. Yeah, he was actually with Michaela Gallagher before he came to the National Institute on Aging. Anyway, what they found was that um, comparing young to aged animals in this particular maze test, the young animals learn to solve the maze more quickly than the old ones. But then there was a lot of variability among the aged animals, and they found they could divide them into, it was useful for subsequent studies to divide the older ones to age impaired and age unimpaired, to try to get at, what these animals are housed in the same environment, same genetics. What explains this difference, which is kind of an intriguing question that hasn't been really solved, I don't think yet. Um, uh, One aspect of the environment that's been studied a lot is stress, mostly from the idea that chronic psychosocial stress, for example, uncontrollable stress, uh, has adverse effects on health, no doubt about that, including brain health. And Bruce McEwen has really done a lot of the important work in this area, the late Bruce McEwen. This is an example. So I mentioned that one of the hallmarks of brain aging is dysregulation of neural network uh, excitability, so that you get hyperexcitability. And so back 20 some years ago, they essentially did like brain dialysis and took extracellular fluid and measured glutamate levels. And they found that as, uh, and then they put the animals under stress. So this is a 1 hour period of restraint stress where they put the animal in like a straitjacket or a tube. They can't move. It's very stressful. Uh, Epinephrine, corticosterone levels go up, the adrenal stress hormone. But they found that, there's some increase in extracellular glutamate in the young animals during the stress and then recovered. The old animals, there's an increase that begins during the stress and it continues afterwards. So there seems to be an impaired ability to keep extracellular glutamate levels low in the aged animals, which means presumably that the receptors for glutamate are activated at a higher level and longer duration than the young animals. Um, right. What did they do here? Huh. I'm sorry, I'm blanking on what they did in this, the difference between the upper and the lower one. Um, it's kind of a similar thing here. I I'm sorry, I'd have to go back, but you don't have to know the details just to say this is consistent with uh, hallmarks of brain aging that I talked about. Now, with functional MRI, where one is essentially measuring blood flow uh, to brain regions. So you're getting an image of relative blood flow. And blood flow is tightly coupled to neuronal activity. So if activity of neurons increases, there's this interesting way through mediation by astrocytes and nitric oxide that you get a dilation of the blood vessels and increased blood flow to that brain region where the neurons are active. It makes a lot of sense because those neurons are going to need more oxygen and energy substrates like glucose. And so this is actually going back to Peter Rapp collaborated with the group at night and did functional MRI and their aged impaired and aged unimpaired animals. So this just shows in particular animals used in this study, the individual animals that were divided into these two bins based on their performance here. And then they looked at functional connectivity um, between brain regions that essentially uh, through this method, one gets an idea of how much neurons in different brain regions are communicating with each other. And there's a reduced functional connectivity in the aged impaired animals compared to the aged unimpaired and young animals. And here's some of their images. Okay, non-human primates. A lot of work on cognition and aging in non-human primates. This shows this, I'm not going to spend much time on this. They're using different tests of learning and memory, and the aged animals um, make more errors and they don't learn as quickly compared to the young animals. And then in this study in the rhesus monkeys, they were interested in the basal forebrain, where there's these cholinergic neurons that send their axons to the hippocampus and other brain regions, but mainly the projections of the hippocampus have been studied in relation to learning and memory. And activity of these cholinergic neurons is important uh, for learning and memory. So if you like kill these neurons with an excitotoxin or other approaches, then that impairs memory, for example. And then, as in all brain regions, there's also GABAergic neurons. So this amino staining with an antibody to choline acetyltransferase, enzyme involved in acetylcholine synthesis, and uh, glutamic acid decarboxylase 67, GAD67 is a protein involved in production of GABA from glutamate. Glutamic acid decarboxylase converts glutamate to GABA. Okay, so they had young and old monkeys, and then they quantified the number of immunoreactive neurons and looked in young and old. So in the total basal forebrain, there's a reduction in oh my God. Yeah, in both the cholinergic and the GABAergic interneurons. And then in this study, they measured the number of parameters of dendritic morphology, essentially total dendritic length, number of dendritic spines, and spine density along the length of the dendrites studied. And this is in layer 3 pyramidal neurons in the cerebral cortex. And what they found in the aged animals, there's reduction in total dendritic length of the apical, but not the basal dendrites. There's a reduction in number of spines in both aspects of the dendrites, and reduction in spine density. Okay, so this is at the cellular level correlating dendritic complexity and synapse numbers, essentially, with and they did cognition in the same animals. All right, I should also point out that there's changes in white matter that occur during aging, decreased myelination, and that can actually be picked up on MRI. Just gross structural changes occur in the brain during aging, thinning of the cerebral cortex. One very obvious thing is enlargement of the ventricles. The lateral ventricle is the main one, and it definitely increases with aging. In Alzheimer's disease, much more dramatic reduction in cortical gray matter and white matter, and therefore there's more room for the cerebrospinal fluid to accumulate because the brain's getting smaller in the skull with defined size. Also, the vascular changes with aging in everyone, pretty much to some extent anyway. And so there's vascular problems that can contribute to age-related cognitive decline and even neurodegenerative disorders. Okay, this is just from a meta-analysis of total brain volume changes with age. And if you look at this overall, if you get beyond 60, it starts to become very clear that there's a reduction in overall brain size, with some relatively more reductions in certain brain regions, particularly frontal and temporal lobes. And then there's a lot of work going on now where groups are trying to establish, estimate an age based on various biomarkers, and then compare that to the actual chronological age of the individual. So the idea is can biomarkers, if you have two people that are, say, 60 years old, can biomarkers be used to estimate or determine whether those two individuals are aging with respect to the brain at different rates? And moreover, in general, if someone's aging at a faster rate than another person, they're probably more likely to develop an age-related neurodegenerative disorder, since aging is a major risk factor for AD, PD, as well as stroke and FTD. Okay, so there's something called brain age gap, BAG. And brain age gap is the difference between the actual age of the individual and the predicted or yeah, if you would, normative age based on biomarkers. And this is from an interesting study a few years ago in eLife, where they looked at cognitively normal. Okay, sorry, I have to back up a little bit. There's now ways you can do PET, positron emission tomography imaging, looking at accumulation of amyloid beta peptide, so amyloid imaging, and tau, in particular pathological tau accumulation. And so in this study, what they did is, so 0 is no brain age gap. So in other words, the individual, if an individual has a brain age gap of 0, that means the pathological tau level is the same as the average pathological tau level for a neurologically normal individual of the same age. Okay. Right. So a negative brain age gap um, uh, indicates, right. Okay. So the way to look at this these are the normal individuals in blue, and they're amyloid negative. So they did amyloid imaging. They had little or no A beta pathology. The green is cognitively normal, positive for amyloid, so they have amyloid pathology. And red is cognitively impaired. So the cognitively impaired humans have um, much higher tau PET signal than these other two groups. So if they're impaired, they have more tau, whether or not they have amyloid pathology. Right. And, oh man, I forgot. It's more complicated than that. They also looked at functional connectivity by fMRI and and uh, show that there's an effect of there's a correlation between tau PET, cognitive impairment, and functional connectivity with aging. Okay. All right. So this slide, just kind of summary of structural changes with age in the brain. This is cortical thickness in blue. The green is total surface area, which declines as the brain shrinks with age. Functional anisopatry is a measure of essentially healthy white matter, which declines with aging. So both gray and white matter declining. And then the white matter hyperintensity is kind of pathological changes in white matter, which increase with aging. Okay, so the last part of my lecture today is going to talk about biomarkers and something biological clocks and focus on the brain, but there's a lot of work outside the brain that's initially done in this area. Um, one thing that's been shown is that DNA methylation increases with aging, and methylation of gene promoters modifies the ability of that gene to be activated by transcription factors. So with aging, increased methylation indicates a declining ability to regulate gene expression to levels in young individuals. Okay, so this has been shown pretty consistently that DNA methylation increases with aging. And this was initially done on white blood cells, but it's also been looked at in brain. And then there's many studies now where DNA methylation is looked at in the same individuals in multiple organs. And then this can be combined with biomarkers of aging. You can do all these omic studies, get literally thousands of measures of proteins, gene expression, mRNA levels, metabolomics in the blood or in cerebrospinal fluid, and then using software, which is advancing quickly, and artificial intelligence. The idea is you can, by having so many different variables, you can identify clusters of variables that predict uh, predict a person's age or predict a person's risk for an age-related disease. This is also being used in cancer field for early diagnosis, and yeah, a lot of work going on this, a lot of interest in this by artificial intelligence companies, actually. Just as an aside, my son recently in January got his PhD from Hopkins. Uh, he was in the biology department, and his work was mainly biochemistry and molecular biology, and so the timing was really bad, as you can imagine. And I'm sure you've talking to your colleagues, this is a very problematic. Hopefully, it's short term, but so he got his PhD, and then all of a sudden, scientists at universities weren't getting any money, grants weren't being funded. So he actually got a job with initially with OpenAI, and now with Meta, helping them develop their AI systems in the realms of biology and medicine and so on. Okay, so that's okay. So what is a criteria for a biomarker of aging? Last time, I think it was last time, I mentioned there's this association between, maybe it was the first one, between low body temperature and lifespan, um, and it holds up in mammals anyway. And they were initially in the Baltimore longitudinal study of aging. That was one of the biomarkers that showed up. And beyond that, there's a couple others, walking speed and grip strength, interestingly. So faster walking speed in midlife predicts longevity, and stronger grip strength. Now, the walking speed, there's a lot of problems with that, because obviously people who have some physical disability, uh, uh, or in general, people with obesity, not always when they're young, they can walk fast maybe, but when you get to mid-age, probably don't walk as fast. So um, there's some, what is actually walking speed measuring. But anyway, here's some of the criteria. This is from pretty review article where they talk about, how does one establish what is a biomarker or biomarkers of aging. And first of all, it has to be correlated with chronological age. It should be predictor of mortality and survival and disease risk, functionality. And one would think it would be related to mechanisms of aging. So some of the biomarkers are hallmarks of aging I talked about last time. And it should respond to factors that accelerate or slow down aging. So in other words, in animals where you can easily manipulate factors, environmental factors, genetic factors, whatever biomarker, putative or proposed biomarker, that should be changed in a way consistent with effects of environmental or genetic manipulations on lifespan. And it should be validated in diverse, genetically heterogeneous populations, and in multiple tissues. Okay, so now there's this integration of artificial intelligence, biomarkers, and molecular clocks, or if you will. And this is kind of interesting because you remember in the first lecture, I talked about whether aging is programmed or not. And the conclusion of most people in the field is that it's it's not. And certainly it's not from the standpoint that environment has a big factors. Uh, uh, but anyway, now there's so what's going on now, and a lot of this is in big groups of investigators studying various populations throughout the world, collecting blood, collecting cerebrospinal fluid, um, you know, from the standpoint of the brain, behavioral data. So, for example, Alzheimer's research centers, for example, they have some interesting programs going on. I was at the University of Kentucky for 11 years before I moved to Baltimore. And they had a really good program there where they would get uh, the spouse, and that's actually fairly common. It's hard to get controls for these studies. There's a lot of people with Alzheimer's, a lot with Parkinson's disease, and ideally one would have age-matched, neurologically normal individuals as controls. And so Kentucky and elsewhere, I think for the most part, uh, they get spouses of the individual that's diagnosed with Alzheimer's and then use them as a control. And that one reason that's good is they're pretty much age matched for the most part. And the other is they live in same environment. They may not always have exactly the same health habits, but actually it's pretty common that spouse partners will have the similar um, environment. And okay, and all of those places, Alzheimer's research centers, Parkinson's, the NIH-funded ones, they have tons of frozen blood, CSF, brain tissue, in some cases other tissues. And so these groups are getting together, do proteomic analysis or other analysis, DNA methylation, for example, and then get all the data together and analyze it. And it makes it much more powerful when some result comes out that's new and interesting. Um, and then the implications for the general public is that someday you may go to your doctor and he will do a panel, take blood, and then a week later you'll get some indicator of your biological age in relation to your chronological age. And on the other hand, we all age, and so I think we can all assume we're aging, and uh, and individuals should want to be able to function well as long as they can, including their brain. So, kind of from a practical sense, I guess it might motivate someone to take action if they see that they're aging a lot faster than they should be. or maybe not. I mentioned extracellular vesicles. This is an emerging area. It's really a lot of work going on in this area now. Okay, so cells can release small vesicles either by the budding of the plasma membrane and pinching off of this and releasing the vesicle, or through this endosomal pathway or pathway involving multivesicular bodies that, as their name implies, they have a bunch of vesicles inside them, and these can fuse with the membrane. So these vesicles can be cargoes that contain mainly cytoplasmic constituents. Uh, some of these vesicles, and these two, have bits of mitochondria in them. Interesting. So you can find mitochondrial proteins in there. These vesicles will have messenger RNA. And this is showing some of the classes of things that these vesicles, there's hundreds and hundreds and hundreds of proteins that have been found in these membrane proteins, some related to immune function, for example, um, uh, transporters inside bits of mitochondria, a lot of different proteins. It can be synaptic proteins. The vesicles are coming from neurons. Signaling cascades, for example, uh, Dimitrios Kapagiannis, a neurologist at NIA who was under me before I retired, he developed a way to isolate vesicles from blood using antibody to L1-CAM, this neural cell adhesion molecule, and pulled down these vesicles and found that they have insulin receptors in them, and that the functionality of these receptors, the ability to activate downstream signaling, is impaired in older individuals with mild cognitive impairment and Alzheimer's, as well as in those with diabetes. So there's, assuming and there's evidence for this, assuming these vesicles are coming from the nervous system or brain, and including the brain, that suggests there's insulin resistance of neurons in the brain. There's other evidence for that. Now, the normal function, these extracellular vesicles seem to have normal functions. For example, they can be released from one cell and endocytosed by another cell and transfer, for example, messenger RNAs. So you can find in the literature many papers now where they have evidence that you can do this in cell culture, like take cells from one tissue, say neurons or astrocytes, microglia from the brain, and incubate them, collect the culture medium, uh, isolate these extracellular vesicles by these centrifugation protocols. There's actually a company that markets a kit where it's easy to isolate total vesicles, and then you can take those vesicles and add them to other neurons or cells from the brain or outside the brain. You can culture cells from the periphery and do that, and so on. You can isolate the vesicles and label them in some way, either with a radio tracer or in some cases antibody, then inject them in the blood or cerebrospinal fluid and see where they go in their brain. And so a lot of interesting stuff going on here. But anyway, as far as biomarkers and aging, here's kind of a protocol for isolating the vesicles, brain-derived extracellular vesicles, using L1 cam or other antibodies, and then you can do all sorts of things with these, as I mentioned. Um, and then there's clinical applications. Um, so in the cerebrospinal fluid and blood, if you just take total cerebrospinal fluid or serum, uh, there you will find some A beta and some tau, and the levels are much higher in patients with Alzheimer's disease. Okay. Also, what seems to be a kind of another maybe less specific indicator of neural damage in the brain is neurofilament proteins, particularly this neurofilament light, lower molecular weight one. So the axons of neurons, of course, they have microtubules. They're loaded with microtubules along which mitochondria are transported, and probably cargoes are transported retrogradely towards lysosomes in the cell body. But the the axons have these neurofilaments, and these can be released certainly if the neuron, an axon is damaged and the membrane's ruptured. Uh, anything in the axon can be released into the potentially in the CSF and potentially into the blood. Um, what's not known is so much or studied, and I think it's actually interesting, the idea that maybe these neurons are normally spitting out cytoskeletal proteins that are becoming damaged, potentially harmful to the cell. So the idea is there is that if you have A beta accumulating or hyperphosphorylated tau, or even there's some problem with the neurofilaments, that those dysfunctional damaged proteins are recognized and exported from the cell in exosomes or by blebbing of the surface. So from the individual neuron's perspective, that may be a way to get rid of something that's potentially harmful to the neuron and letting other cells, for perhaps microglia, take up and or astrocytes take up and degrade those molecules. I just want to point out, because this is often ignored, there's some normal level of, in this case, neurofilament light in the blood. This looks at aging, and one indicator that I may be right about this is look early on, the neurofilament light levels are higher in kids. Like I have one granddaughter now who's 2.5 years old, right? So, well, maybe we're close to the same now. So may have one thing in common, our serum neurofilament light. But when you get into when you get past, and if you remember, during this early period of human development, the prefrontal cortex is not yet fully developed, and in particular, you have a lot of axons that are not yet myelinated or fully myelinated. So the myelination particularly in the frontal lobe, uh, but maybe other regions, is still going on. So there maybe these uh, axons are spitting out more NFL during this period. That's kind of speculation, but it's consistent with what I've said. Okay, and then you get kind of stabilization during the uh, formative years, most performative years, where you're most creative and so on. So you guys that are listening to me, you're much more creative than us older folks, and you got more physical and cognitive abilities. So take advantage of it. But get enough sleep. Wait for my last talk, because you need to challenge yourself mentally, physically, but you have to have recovery period every day. enough sleep. Okay, but anyway, this NFL is being kind of used as a marker of neural damage and dysfunction during aging. Yeah, so I mentioned that these extracellular vesicles have insulin receptors in their membrane, and the insulin receptors, uh, they have a lot of proteins that associate with the receptor, as is true for a lot of receptor proteins. And one thing that happens very rapidly when insulin binds to its receptor, so I'm talking about within minutes, is translocation of the glucose transporter GLUT4 from the cytosol to the membrane. So there's more transporter in the membrane available to take up glucose from the extracellular fluid, the blood, and get it into the cell where it can be used to generate ATP. But one of the proteins that associates with the insulin receptor is called insulin receptor substrate 1, IRS1. And it's positively coupled to the mTOR pathway. All right. And so this is kind of interesting because on the one hand, chronic activation of the mTOR pathway is not good, but transient activation, as long as there's a recovery period, is good. And I should also say the insulin-like growth factor receptors are positively coupled to mTOR. Okay, but anyway, this IRS1 can be phosphorylated on both serine and threonine residues and tyrosine residues by serine threonine kinases or tyrosine kinases. And it turns out there are antibodies that have been developed that only recognize IRS1 when it's phosphorylated on particular serine, that's one antibody, or tyrosine, a different antibody. And phosphorylation, right? Phosphorylation on the serine is an indicator of reduced or impaired, what is it that way? Yeah, reduced or impaired signaling of the insulin in the insulin pig pathway, whereas phosphorylation on the tyrosine residue indicates an active pathway. So there's a study done, published 10 years ago, where they used the antibodies to phosphoserine 312 and actually a pan-tyrosine IRS1 antibody. And they looked in, and this is total IRS1, phosphorylation independent. Looked in aged controls, Alzheimer's disease. Right, so this is control, Alzheimer's disease. This is frontotemporal dementia control, and then patients. And this is diabetes control for the diabetes group. So anyway, what you see is with the Alzheimer's disease and the diabetes individuals, extracellular vesicles isolated from the brain have reduced tyrosine phosphorylation, which indicates reduced activity in the pathway. Anyway, that's correlated with this insulin resistance index, which is a ratio of the phosphoserine 312 to the phosphotyrosine. Okay, so I'm almost done for today. Oh, I should say one thing. Well, I'll say it at the end, actually. Um, right, so we talked about inter-individual variability, the fact that genetics accounts for, depending on the study, no more than 30% of aging trajectories um, and sporadic uh, disease. Uh, uh, Alzheimer's disease, there's some, the ApoE4, for example, is a risk factor, but it's uh, uh, not uh, causal. So there's a lot of work looking at the exposome, the various environmental factors that individuals are exposed to, whether it's pollution, your particular community environment, uh, climate change, your lifestyle habits, and so on, and level of education. There's so many different environmental factors that can be incorporated into studies of aging in epidemiological studies, but then also in clinical trials. It's really hard to look at aging, but if you can develop biomarkers of aging that are pretty reliable, then you can ask whether some intervention over a period of, say, 6 months or a year uh, changes uh, in response to some manipulation of the environment. A lot of work going on with exercise, dietary interventions, uh, cognitive training, and other things. Right, and I think this is my last slide. So I mentioned this idea of bad stress and chronic stress that accumulates over time. It could be psychosocial stress. It could be some chronic physical injury, chronic pain, for example. Um, it could be something like have well, we have even maybe less control over is what we're breathing and being exposed to in the water. Apparently, we all have these small plastic beads in our brains. Don't drink out of the plastic bottles, particularly if you leave them in heat. Um, I think that's the way things are emerging. Okay, but there's also good stress, and which I'll talk about. Exercise is a stress during the exercise. Fasting, energy restriction is a stress. Um, Using your brain is a stress on your neurons. Calcium, sodium calcium influx, increased free radical production, uh, you know, increased actually oxidative damage when you're using your neurons, but as long as you give them a chance to recover, they're actually better off for having experienced the stress of exercise. Okay, so I'm going to stop there and if I can find sharing.